Okay, so now we're going to talk about some human cases where the number of chromosomes is going to be incorrect. And the first we'll talk about is the most common, and that is Down syndrome. It was discovered, or at least characterized, in 1866 by a man called Langdon Down. And this is known as trisomy 21. And what happens here is you'll have 3 copies of a chromosome 21. So that's why it's called the trisomy. In terms of genetic designations, we're going to go through several of these. So the way that it's designated here is 47, because there's 47 total chromosomes, and a 21 with a plus after it, meaning the chromosome 21 is the one that is the extra copy. This is the only human autosomal trisomy in which a significant number of individuals survive longer than a year past birth. And you might expect, although this additional chromosome should be equally derived either from the mother or the father, in this case, the egg happens to be the source in about 95% of the cases of Down syndrome. So many common techniques have been used to diagnose many of these different genetic disorders through prenatal testing. Amniocentesis happens when fetal cells are obtained from the amniotic fluid. The chorionic villus sampling, or CVS, happens when fetal cells are obtained from the chorion of the placenta, so an actual part of the placenta. Newer techniques are called non-invasive prenatal genetic diagnosis, and the cool thing about this is the fetal cells and DNA can be derived directly from the maternal circulation. So you can just go take a small blood sample from the mom, and there's no risk to the fetus. In contrast, the amniocentesis and CVS themselves, just the procedures, have a very high rate of miscarriage or problems in the developing fetus. And so if we can use these non-invasive techniques, the prognosis will be much better for these fetuses. Now you can see here the rate of Down syndrome per 1,000 births as compared to the maternal age. So at a young maternal age, going up through about age 30 is about 1 in 1,000, increases to about 3 in 1,000 at age 35, jumps up a little bit to 10 in 1,000 or 1 in 100 by age 40. So people talk about this skyrocketing risk after, age 35 for women to have children, and surely, yes, it does increase about tenfold between age 30 and 40, but we're still talking about a pretty relatively small number. 1% of individuals at age 40 might have a child with this Down syndrome. Now, if you're talking much beyond that, however, by about age 45, it's a 1 in 30, and by age 50, it's about 1 in 15. So you can see after about age 40 is where I would say there's a really strong drop-off, really strong increase in the number per birth. So some other cases that we have for different trisomies and monosomies, most of these do not survive to term. And in fact, most of these are what medical term for miscarriages is spontaneous abortions. So the monosomy with the highest incidence rate causes Turner syndrome if the fetus survives to term. This is designated as 45 with an X. So it only has 45 chromosomes instead of the 46 chromosomes. The chromosome affected is going to be the X chromosome. So these are going to be females with only 1 X chromosome. A few other autosomal monosomies are going to be very rarely found. You really should expect relatively equal frequencies of the monosomy and trisomy, right? If the non-disjunction's happening, you'd expect equal proportion of the gametes to be monosomic and trisomic. We really don't know why this is. Some suggestions or thoughts. One is that the gametes lacking a single chromosome might be functionally impaired. Maybe they just don't work very well. Maybe the eggs aren't released, or maybe the sperm aren't released, or maybe this is just some other problem that we don't know about yet. It may also be that a conception may occur, but the embryo may die too early, so that the recovery occurs infrequently. Maybe women don't even know that they're pregnant at this point. Maybe it just happens so early, and so you weren't able to actually recover any tissue to be able to do any of the sequencing or analysis. There are 2 other human trisomies that survive to term. However, it's primarily lethal by about age 1. And these are known as Patau syndrome. And again, it's 47 chromosomes instead of the 46. The 13th chromosome is the one that those patients have in addition. And Edwards syndrome, 47 chromosomes, and the 18th chromosome is the one that there's going to be 3 copies of. Both of these, they're going to have very severe abnormalities, early lethality, a lot of problems with brain development, a lot of problems with kidney, and other problems like that.